All right, so you're here because you want to get into the nitty-gritty, right? You want to really understand something complex, and you want to do it quickly.
Absolutely.
We hear ya.
And today, we're going to take you on a journey deep inside a tooth. We're talking root canals.
Oh, yeah.
We're going deep into a scientific study that takes us right into the action, showing us what happens when a dentist starts cleaning out an infection.
Yeah, this is pretty cool. This study looked at 15 single-rooted teeth, and they all had those periapical lesions,
Yeah.
that damage that happens at the tip of the root because of the infection.
Right, right.
And the really interesting thing is that the researchers really focus on just the mechanical cleaning part of the treatment.
Okay, so no fancy antibacterial potions yet.
Nope, just saline. They wanted to see what just the physical cleaning could do on its own.
I see. So it's like we're isolating that one variable to see its impact. And how did they actually track what was happening in there? Well, they took bacterial samples at different stages of the treatment. Yeah, really smart.
Yeah.
That way they could see how the types and the number of bacteria changed as the cleaning went on.
So we're really getting a microscopic view of the battlefield, so to speak.
Exactly. The microscopic view of what's going on inside a procedure that, let's face it, so many people have experience, but very few truly understand at this level.
All right, let's zoom in then. Before any cleaning happens, what's the situation inside those infected root canals? What kind of bacterial party are we crashing?
Well, unsurprisingly, every single tooth had bacteria in its root canal. I mean, that's why they needed the root canal,
Of
right?
course.
But the numbers were really striking. The middle ground, what we call the median, was about 400,000 bacterial cells.
Whoa, that's a lot of unwelcome guests.
It is. But some teeth had even more, like way more. We're talking 10 million in some cases.
10 million, that's like a whole city packed into your tooth.
Yeah, microscopic city of troublemakers. And it wasn't just one type of bacteria either.
Oh, so I'm going to ask about that. So it's not just a monoculture in there.
No, not at all. Each tooth had somewhere between 1 and 10 different strains of bacteria.
Wow, so quite the diverse community.
Yeah. And here's the thing. Most of them were talking 88% were anaerobic.
Anaerobic. That means they don't like oxygen.
Right, exactly. They thrive in that oxygen-core environment inside the root canal.
Right, they've adapted to that little world down there.
And the study actually named the most common culprits, the usual suspects you find in these initial infections. We're talking Peptostreptococcus micros, Peptostreptococcus anaerobius, Fusobacterium nucleatum.
Okay, we get it. Lots of tongue-twisting games.
Yeah, those are just a few of the big ones, but they give you a sense of who's causing the trouble in there.
Right, a whole gang of oxygen-hating microbes. So the scene is set. Now the dentist starts cleaning. What happens to this bacterial population when they start filing and flushing with saline?
Okay, so this is where it gets interesting. The main takeaway is that the mechanical cleaning really does make a difference.
Okay, good to know.
The researchers found a significant decrease in the number of bacteria after each appointment. We're talking a reduction of 1,000 to 10,000 bacterial cells compared to the start of each cleaning session.
So even without any antibacterial agents, the physical cleaning itself is disrupting their little world.
Exactly. It's like shaking up the whole ecosystem.
Were there any cases where the bacteria just gave up after the first cleaning, like they couldn't handle the disruption?
You know, there were actually 3 cases where that seemed to happen.
Really?
Yeah, the bacteria found after the first cleaning just didn't reappear in the next appointment.
Huh. So sometimes just that initial shakeup is enough.
It seems so, at least for some types of bacteria. In one case, it was just Streptococcus meteor that disappeared.
Okay.
Another case, it was Solanomonas butygena that didn't come back.
Interesting.
And in the third case, it was a mix of Eikenella corrodens, Bacteroides esacrolyticus, and a type of Fusobacterium they couldn't quite identify.
So it seems like different bacteria have different levels of resilience, or maybe just bad luck.
Yeah, it's a fascinating insight into the dynamics of those microbial communities.
Absolutely. But let's zoom out a bit. How often were they able to completely eliminate the bacteria from the root canals using just this mechanical approach?
Well, the good news is that in 8 out of the 15 teeth, all the bacteria were gone after the full course of treatment.
Okay, so a little over half the cases were completely cleared.
Right, and remember, that was after 5 separate appointments, each with mechanical cleaning and saline irrigation.
So not a quick fix, but in those cases, the mechanical approach was ultimately successful.
Yeah, which is pretty encouraging, to see that just the physical cleaning can be that effective.
Yeah, it's a testament to the skill of dentists and the tools they use.
Absolutely.
But that also means that in 7 teeth,
Ah, yes. In those 7 teeth, The bacteria were more stubborn. They persisted even after all 5 cleaning sessions.
So even with the best efforts, sometimes those infections can really hang on.
Exactly. It highlights just how challenging it can be to fully eradicate those deep-seated infections.
Did the study give any clues as to why those particular infections were so persistent? Like, were there certain types of bacteria that were just really good at surviving?
Well, this is where it gets really interesting. The study found no evidence that specific types of bacteria were responsible for those persistent infections.
Really? That's surprising.
I know, right? You might think that some bacteria would be inherently tougher, harder to kill.
Yeah, like they had some special armor or something.
Exactly. But that wasn't the case here. They couldn't pin the persistence on any particular bacterial bad guy.
So if it wasn't the type of bacteria, what was it? What made those infections so hard to eliminate?
The main factor they identified was actually the initial number of bacteria.
Oh, so it was more about the size of the invading force.
Yeah, it seems so. The teeth where the infection persisted tended to be the ones that started out with a higher number of bacteria.
So the more bacteria at the beginning, the harder it is to completely eliminate them.
Exactly. It's like trying to clear out a crowded room versus a room with just a few people.
Right.
And it also highlights the importance of early intervention.
Oh, yeah, definitely.
The sooner you can address the infection, the smaller that initial bacterial load is likely to be, and the better the chances of completely wiping them out.
Now, you mentioned earlier that the types of bacteria in the root canal could actually change during the treatment.
Oh, yeah, that was another interesting finding. Even in those cases where the bacteria persisted, the proportions of different types weren't static.
So it wasn't just the same group holding out.
No, the makeup of the community could shift. You might have one type dominating at the beginning, then after cleaning, another one might become more prominent.
Huh. Almost like they're competing with each other.
Yeah, exactly. And in those persistent infections, they also found that the bacteria often rebounded in number between appointments.
So even after being knocked down by the cleaning, they could multiply again when left alone.
Right, especially when the canal was sealed up, it provided a protected environment for them to recover.
A temporary reprieve, but not total defeat.
Exactly. And there's actually a table in the study, Table 2, that really illustrates this dynamic.
Oh, so we can actually see these changes happening over time.
Yeah, it shows the percentage of different species across all 5 appointments in a case of persistent infection. You can see how, for example, anaerobic streptococcus species might be a big chunk of the population at the start, then drop after cleaning, but then increase again by the next appointment.
A real back and forth battle.
Exactly. And you see similar fluctuations with other species like Esmidier, various types of Peptostreptococcus, Lactobacillus, Eubacterium millentum, and different bacteroids and Fusobacterium species.
So it really emphasizes that this is a dynamic process, not just a one-time event.
Exactly. It's a constant push and pull between the cleaning and the bacteria trying to reestablish themselves.
So to bring this all together, what did the study highlight as the most important factors for success when using just mechanical cleaning?
Well, as we've discussed, the initial number of bacteria was a big one. The more you have at the beginning, the less likely you are to completely eliminate them with just the mechanical cleaning.
Okay, so early intervention is key.
Definitely. They also pointed out that in long-standing infections, the bacteria might have had time to burrow deeper into the tooth.
Deeper how?
Like into the dentinal tubule?
Dentinal tubules.
Yeah, those are tiny little channels within the tooth structure, and also into accessory canals.
Accessory canals? What are those?
They're like little side branches off the main root canal. And these areas can be really hard for the instruments and even the irrigation to reach effectively.
So it's like they're hiding out in these microscopic bunkers.
Exactly. It makes it much tougher to completely dislodge them.
Now, the periapical lesion, that area of damage at the root tip, did they find that it was a source of bacteria that could reinfect the canal?
You know, that's a good question. And surprisingly, they didn't find strong evidence for that in most cases.
Huh. So it's not like a reservoir of bacteria waiting to strike back.
It seems not. They found that the bacteria were generally concentrated within the canal system itself, or just in the tissues right next to it, not deep within the lesion or the bone.
Okay, so that's actually good news in a way.
It is. It means that focusing on thoroughly cleaning the canal system is likely to be the most effective approach.
Now, they only used saline for irrigation in this study.
Mm-hmm.
Did they think that might have had any antibacterial effect itself, even though it's not a dedicated disinfectant?
Well, they did acknowledge that possibility.
Oh.
They said that some microorganisms might not be able to survive in saline for very long periods.
So just being in that environment might be detrimental to some of them.
Yeah, but they pointed out that the contact time during this study was pretty short.
Right, it's not like they were soaking in it for hours.
Exactly. They also mentioned that exposure to oxygen during the treatment could have a slight effect on the bacteria.
Because most of them are anaerobic, right?
Right. Even that brief exposure might be enough to disrupt some of them.
Interesting. And what about the temporary filling material they used? Could that have had any effect?
They considered that too, but they said they designed the study to minimize any direct contact between the filling material and the canal space.
So they tried to isolate the effects of the mechanical cleaning as much as possible.
Exactly. They wanted to be sure that any changes they saw were really due to the cleaning itself.
Now, how do these findings compare to what other researchers have found in the past?
That's an important question. There have been other studies that looked at using just saline irrigation for root canals,
Okay.
and some of those studies reported getting negative bacterial cultures, meaning they didn't find any bacteria after just the first appointment.
Oh, wow. That's pretty impressive.
Yeah, they were finding that in about 20% to 30% of cases.
Huh?
But this study, they used really sensitive methods for detecting anaerobic bacteria,
Oh,
and they didn't see those high rates of elimination after the first cleaning.
interesting. So maybe those earlier studies weren't catching all the bacteria.
It's possible. The methods for detecting bacteria have improved over time, so what might have seemed clean in the past might not be considered clean by today's standards.
So it's a reminder that technology and techniques are always evolving.
Absolutely. And as for long-term outcomes, without using antibacterial dressings between appointments, other studies have reported a wide range of results.
Okay, so no clear consensus.
Right. Some studies found no bacteria at the second appointment in any of the cases, while others found bacteria in almost half the cases.
Wow, that's a big difference.
It is, and some researchers have suggested that the specific types of bacteria present initially might influence those outcomes.
So some bacteria might be inherently harder to get rid of.
That's the idea. But, as we discussed, this particular study didn't find support for that idea.
So it seems like the initial bacterial load might be a more important factor than the specific types of bacteria.
Yeah, at least based on this study's findings.
So the overall message from this study seems to be that while mechanical cleaning is essential, it's often not enough to completely eliminate the bacteria,
That's a good summary.
and that the initial number of bacteria seems to be a major obstacle.
Exactly. They really emphasized that point.
What was the main conclusion the researchers drew from all this?
Well, they concluded that while mechanical instrumentation and saline irrigation are very effective at reducing the number of bacteria in an infected root canal, they often don't eliminate them entirely.
Okay.
And that this suggests that using disinfectants, like antibacterial irrigating solutions and medications placed inside the canal, is probably crucial for long-term success.
So the mechanical cleaning is like the first strike, and then the disinfectants come in to mop up the survivors.
Yeah, that's a good analogy.
All right, so for those of you listening, here's the takeaway. When you get a root canal, the cleaning your dentist does is super important. It dramatically reduces the number of bacteria, and sometimes it can even eliminate them completely. However, if there's a large infection at the start, some bacteria often survive. And interestingly, this study didn't find that specific types of bacteria were the main culprits in those persistent cases. Instead, the initial number of bacteria seems to be the bigger factor. This really underscores why those additional disinfection steps are so critical for success in the long run.
That's a great summary, and I think this study does a fantastic job of really showing us what's happening at the microscopic level during a root canal.
Absolutely. It gives us a whole new appreciation for the complexity of this common procedure.
Yeah, and it highlights that even procedures we think we understand well can still hold surprises when we really delve into the details.
So here's a final thought to ponder. This study focused on the mechanics of cleaning and the need for disinfection. But what other factors might be at play here? Could there be something about the way our bodies respond to the infection, or the way bacteria behave within the intricate anatomy of the root canal that we haven't fully explored yet? What about the materials we use in these procedures? Could those have subtle effects that we're only just beginning to understand?
Those are all excellent questions, and I think they remind us that even in a field as established as dentistry, there's always more to learn.
Absolutely. And those unanswered questions are exactly what drive continued research and innovation, leading to better treatments and outcomes in the future.
Couldn't have said it better myself.
And on that note, we'll wrap up this deep dive. We hope you found it enlightening, and we'll see you next time for another exploration into the fascinating world of science and health.
Until then, stay curious.
Yeah, keep learning.
Exactly.
Bye.
Bye, everyone.